Welcome everybody! This is going to be our first video lecture for microbiology. I will be making these for every chapter. The chapter will be broken down into smaller segments because, nobody wants to listen to somebody drone on for an hour. Take good notes to study from. These presentations do correspond to your book chapters. You can go back into the chapter to supplement your notes. As we get started, I'd like to suggest a way to study for this class. First, before you listen to the lecture, you should skim the chapter in the book. Look at the section headings, read a few sentences from each section, examine the figures, and read the legends. This will prepare your brain to better absorb the material in the lecture because you are now attuned to the topic. Next, listen and take notes for each chapter. Then go back and read the chapter in more depth. But you can skip any sections that are not covered in the lecture. Use the book to supplement your notes. Look more closely at the figures and be sure you understand what they are trying to convey. Do your homework and the Learning Catalytic Self-Test, which will be in the Mastering Microbiology tab in Canvas. Of course, you should participate in the planned study groups as well. You can visit Mastering Microbiology and watch the videos and animations that go with the chapter. If you do this as we progress and not wait until right before the exam, you will be better prepared to study before the exam. Now let's begin chapter 1. This covers the history and overview of the field of microbiology. There are some big names in microbiology that are covered with their main contributions in the field. The early years of microbiology were really just ones of discovering that microbes existed. One of the important people for this era in microbiology was Antony van Leeuwenhoek. His story is really interesting. He was a cloth merchant, meaning he sold fabric for a living. He also had a special skill. He was able to make very good, very powerful lenses. He put them into little microscopes. Really, they were just powerful magnifying glasses, but they were really good. The thinking is that he initially was making lenses so that he could take a look at the cloth he was buying to make sure he was, paying a fair price, that the cloth was of high quality. You've heard of thread counts in sheets, right? The higher the count, the better the quality. Well, this is the same type of thing. He was taking a good close look at the fabric to examine the quality. Leeuwenhoek did not stop looking at cloth. He was a curious individual and he started to turn his lenses to look at the rest of the world. He realized that there were these microscopic animalcules. That was his term for them. There were these little animals all over the place. Leeuwenhoek, he was the first to describe the world of microbes. So let's think about this for a minute. Where do you think he first saw these microorganisms? A drop of blood on a flake of skin, a splinter of wood, maybe a drop of water? What do you think? If you said a drop of water, you're right. It's not uncommon for instructors to show the microscopic world to curious students by having them look at a drop of pond water. There is so much going on in a productive drop of water. There's this whole little world, an entire little ecosystem that's going on in there. This next slide is showing you an image of the microscopes, the powerful magnifying glasses that Leeuwenhoek was making. You can see the little lens right there, and the specimen would balance on the little pin, which could be adjusted with the screw over here, and it would bring it into focus. This image is showing you some of the different types of microscopic life that Leeuwenhoek first observed. Well, what do you think the Academy of Sciences thought about his initial letters to them? The letters describing these living forms that he saw in a drop of water. If you guessed that they thought he was a little nuts, you'd be right. But he kept writing these letters describing the microscopic things. Eventually, these copious letters convinced the Academy, that is, it convinced their own scientists to take a close look at the things he was describing. Sure enough, when they were able to get a lens of high enough quality in their microscopes, they could see the same things he was describing. So Leeuwenhoek became very famous for these initial descriptions. He wasn't even a scientist in training. If you stop for a minute and take a look at this, you're going to realize that the microbes that Leeuwenhoek was describing fall into the categories that we use today. Leeuwenhoek described what we now know to be prokaryotes. These are the bacteria and the archaea. These two groups of prokaryotes, which actually consist of the two of the three domains of life, are really quite diverse. And we'll describe these in more details later. Leeuwenhoek also saw fungi, protozoa, algae, and small multicellular animals. You might find it interesting that we name microorganisms in the same way that we name other organisms. Carolus Linnaeus, who was a Swedish scientist, developed that taxonomic system. The system today is organized from domains, kingdoms, phyla, order, families, all the way down to genus and species. Microorganisms are grouped the same way. They too are designated by a genus and species. When you hear E. coli, you are hearing the abbreviated genus E, which stands for Escherichia, followed by the species name coli. Now we'll just start describing some of these groups more broadly. The first group, the prokaryotes, as I mentioned, consist of the bacteria and the archaea. These types of living cells are unicellular and they do not have nuclei. The cell does not have a nucleus. These are simple cells. In a later chapter, we will talk about the cell structure in detail. In general, they tend to be much smaller than the eukaryotes. Those are the organisms with a nucleus. They're found anywhere there's moisture, so anywhere on Earth there is available water, you will find prokaryotes, including in the very extreme environments such as very, very hot, very, very cold, and very, very salty. They do reproduce asexually. They just basically double everything and divide. For the prokaryotes, there are these two groupings, that the bacteria and the archaea. Bacteria you've probably heard before. Maybe you've heard about archaea too, but perhaps not yet. They are thought to be the more ancient form of life, and they are found in the very extreme environments. They're both prokaryote. You might be asking, what is the difference? There are several structural differences, but one of the easiest ways to differentiate them is by whether or not their cell wall contains peptidoglycan, a specific molecule we will examine in Chapter 3. Bacteria almost universally contain peptidoglycan, and the archaea contain other molecules in its place in the cell wall. There's a variety of other polymers that they can use, but they don't use peptidoglycan. Importantly, they both don't have a nucleus or any of the organelles of the more advanced cells. If you look at the members of these different groups, the DNA sequences are really quite different from each other. So they're really not that closely related, even though they're both prokaryotic. We're going to spend more time with these later. This next image is interesting because it allows you a visual of the scale of these different types of organisms. Here we see two epithelial cells. They're like your cheek cell. They obviously have nuclei, but if you look, attached to them are a whole bunch of bacteria. These bacterial cells are much smaller than eukaryotic cells. In general, this is true. The typical prokaryotic cell is very much smaller than the typical eukaryotic cell, as shown here. Well, let's look at a couple other categories of microbes. The fungi belong to the eukaryotes. All the rest of the organisms we'll talk about, except for the viruses, belong to the eukaryotes. So the fungi are eukaryotic. They have to obtain food from other organisms. They do possess cell walls, but their cell walls are different than bacteria. In the fungi, they have different types of cell wall components, maybe chitin, maybe cellulose, but different types of cell wall material compared to the bacteria. They're divided into two major groupings. First, the molds, which are multicellular. They have hyphae, which are those little thread-like extensions that you see coming out over here. When you have a whole bunch of hyphae, it forms that visible fluffy mass called a mycelium. If you've ever seen a moldy cheese or moldy bread and you see that fuzzy little structure on the surface, well, that's a mycelium because it's visible. But if you looked under the microscope, you would be able to see individual hyphae that compose the mycelium. Individual hyphae are too thin to see with your eyes without the help of magnification. Fungi can reproduce both sexually and asexually. While molds are multicellular, the yeasts are unicellular. Yeast reproduce asexually by a process called budding, as seen here. We'll talk more about molds and yeast and a little bit more about their reproductive capacity in the laboratory. Protozoa is the next group. These are the single-celled eukaryotes. There's a wide variety of protozoa, and they tend to be divided by the way they move or their lack of movement. If they move by pseudopodia, as shown here in this amoeba, then they belong to that group, the amoeboid group. If they move by the short hair-like structure called cilia, well, they're ciliates. If they move by flagella, then they're a flagellate. There's a fourth group of protozoa that don't move at all. They belong to the sporozoans. Algae is the next group of eukaryotes that are microscopic, at times at least. We put them in this grouping even though they might not always be microscopic. They might be unicellular or they might be multicellular. The unicellular ones are definitely microscopic, but the multicellular ones may be visible to the naked eye depending on the organism. Importantly, algae all are photosynthetic. They do reproduce with some simple reproductive structures, and they can be grouped even further on the basis of the pigments they have. The two examples in these figures below are ones that we're going to observe in the lab. This happens to be a filamentous alga called Spirogyra, and these are photosynthetic, or at least the shells of photosynthetic organisms. The shells make them quite beautiful and interesting. These are diatoms. This next type of organism is considered a small multicellular animal. Some of the parasites that we will be talking about, the things like tapeworms and flukes and pinworms, they're actually small multicellular animals. They're grouped under microbiology because part of their life cycle, if not the entire thing, is going to be microscopic. Here's an example of a small worm that is amongst the blood cells here in a blood smear. The last group of microorganisms is not really an organism, but we study it in here. It's the viruses. A virus is a simple structure, really. We're going to spend an entire chapter looking at them, and I'm sure we'll be talking a lot about viruses through this semester because it's relevant to this time we're in. This image is a virus that attacks E. coli. A virus that attacks bacterial cells is called a bacteriophage. You can see these here. This is the last group of microorganisms that we will be talking about during the semester. Leeuwenhoek didn't see them though, they're too small. So that's just the thought question for a minute. Let's see if you're still awake. Did you hear what I said earlier? What habitats do you think you might find archaea in? What do you think? Where might you find archaea type organisms? Hot springs that are acidic, swamp mud, the Great Salt Lake, or all of the above? Okay, if you guessed all of the above, you are correct. The archaea exists pretty much in every type of environment that you can think of, but we do find archaea in the more extreme environments where we don't find bacteria. All of these things can be considered somewhat extreme, right? The Salt Lake, salty. Swamp mud is very anaerobic. Acidic hot springs is very much an extreme environment of low pH and high temperatures. Archaea exist in all of them. Not the same species, but different types of archaea exist in all of these different environments. All right, this is where I'm stopping this first video, and I will pick up with part 2.